So we've had 2 days in which we've learned everything there is to know about molecular replacement. So what I want to tell you about now is how we can use some of these tools in a specific case of coiled-coil proteins. So why are we interested in coiled coils? Well, the interest of the lab is meiosis. Okay, and in meiosis, the sort of whole point is to halve the chromosome number down to 23 and exchange the genetic material to create genetically diverse haploid germ cells. And this happens in the cell in a very involved way, in which double-strand breaks are induced and are repaired by the homologous recombination pathway, but using the homologous chromosome as the template. And the point of that being is that chromosomes get dragged around the cell so that these double-strand breaks can find the matching sequence in the homologous partner. And then once recombination intermediates are formed, they act as physical tethers that connect together the homologous chromosome pairs where the sequences match. And then once this happens a number of times, these local alignments get converted into one complete alignment between the homologous chromosome pairs by assembly of this the synaptonemal complex. The synaptonemal complex is essentially a massive molecular zipper, a protein zipper that just zipped together the two chromosomes along their entire length. And this structure was first reported in the 1950s. Sort of it's a classic sort of meiosis structure. It's absolutely essential, and it provides the sort of 3D environment for the repair of these recombination sites and the formation of crossovers. So the main focus of the lab is to work out the 3D structure of the meiotic synaptonemal complex. So the synaptonemal complex contains 8 proteins that we know of so far, and they are all essentially coiled-coil proteins. So that's why we are interested in solving coiled-coil structures. And the one I'm going to show you as an example today is SYCP1. And SYCP1 provides the sort of underlying framework of the synaptonemal complex. It's a long coiled-coil protein. Its N-termini interact in the midline, and its C-termini self-assemble on the chromosome axis. So it sort of provides the teeth of the zipper, as it were. So what I just want to first say is that coiled coils are quite an unusual case. They're possibly the most well-understood protein fold, if you think about it, because it is just helical structures binding together in a coiled fashion. But they're extremely diverse. So here are an example of all sorts of ones that you can find. And they're involved in pretty much every cellular process. It's supposed to be about 10% of proteins contain coiled-coil regions. And given the asymmetrical, sort of an elongated shape, they often act as molecular rulers or rods, ropes to hold things together, or scaffolds for larger assemblies to form within the cell. And you can, in this diversity of structures, you can really split it up into 2 simple features. The oligomer, because you can form a coiled coil of 2 helices, 3, 4, 5, you can form massive coiled coils, and whether they are parallel or anti-parallel structures. Those are the 2 sort of simplest statements. And the thing is that understanding the oligomer and whether something is a parallel or anti-parallel coiled coil tells you a lot about the biological system that you're thinking about. Because if you have 2 large domains that bind together through an anti-parallel coiled coil, it's pretty obvious you get 2 large domains with a big rod in the middle. So they really do tell you everything you need to know. But these are very difficult to predict from the sequence, which is why we want to solve crystal structures in the first place. The other thing to point out is that coiled coils have a lot of more subtle diversity within the structure. So the pitch angle between helices will vary quite a lot, as you can see here. you get kinks, gaps, turns, and all of these things essentially mean that even if you know you're dealing with a certain type of coiled coil, you have a homolog, molecular replacement is likely to fail just because of little minor differences between those structures. So they do pose a particular challenge for a molecular replacement. They pose additional challenges too in terms of the crystallography. So coiled-coils are long and thin, so by definition like to form crystals that look a bit like this. This is the sort of common type in which they all just sort of stand up next to each other. And you often get quite weak contacts laterally, so you get a lot of crystal defects. You can see some pretty weird diffraction patterns from coiled-coil crystals, and you typically get quite anisotropic data. And I mean, you can see here, so you classically get spots that are close together that are from the long axis, as in this axis, and then spots that are further apart in this direction that come from this sort of lateral association. And you typically get better diffraction along the axis. So this direction will typically go to a higher resolution, and then this direction is typically poorer as well. So this is a feature that commonly comes out. They show translational non-crystallographic symmetry frequently, and you can see here you can get translational NCS from sort of here to here. This can be true translational symmetry. It can also be false, because a coiled coil that way around looks a bit like a coiled coil that way around. So often you get translational non-crystallographic symmetry that isn't actually true because it's back to front. And you can see this in a native Patterson map of a coiled coil data set, where you get these this pseudo-centering vector there. You get additional translational NCS there. But the Patterson map is actually dominated by these additional features. You see these ripples here. All of these ripples are 5.1 angstroms apart. And these come from self-Patterson vectors just from the coiled coil itself. The coiled coil has a repeat of 5.1 angstroms. And so that's what we're seeing here. So these Patterson features clearly are going to have a negative effect on trying to use a molecular replacement, and they are clearly going to mask the subtle features that you would otherwise want to pull out of a native Patterson map. Now, alongside this, I've already pointed out about conformational changes and structural alterations between different coiled coils, but also, you can just ask the question, why not just make the selenium methionine protein? It's actually often quite tricky in this case. Often there aren't enough methionines. If there are enough methionines, we've seen quite frequently the protein is suddenly not soluble anymore, because presumably they're at the interface. And even if that all works, these native Patterson features often make finding heavy atom sizes tricky. So there is an advantage if we can try to get around this with molecular replacement instead. And there are advantages. So I've given you the downsides of coiled coils, but there are advantages of coiled coils as well, which is that they are entirely helical. And as we've already heard, helices have a characteristic appearance in electron density maps, and it's typically sort of easier to build helices into pretty bad maps. And obviously, we have a well-characterized helical structure, so rigid little fragments of helices make for very good search models. And these features have been exploited in a number of programs, most notably ARCIMBOLDO and AMPLE, which you've heard about both of them. And I've just sort of put up here that in the last few years, we've solved all of these coiled-coil structures, and only one of them required experimental phasing. So you can typically solve coiled-coil structures using the advances in ARCIMBOLDO and AMPLE to get around having to do experimental phasing. And I'll also point out that almost all of these are within the sort of 2.5 angstrom range. So we're not talking about having ludicrous data. We're talking about having sort of pretty average data. So I'm going to illustrate this with two examples from the same project. So I said about how SYCP1 forms the sort of main protein of the synaptonemal complex. Well, we crystallized its C-terminal end, which self-assembles on the chromosome axis, crystallized it in two different crystal forms. So one had a slightly better resolution, but it had 4 chains in the asymmetric unit. The other had a slightly worse resolution, but only had 1 chain in the asymmetric unit. So each of these has a slight sort of advantage over the other. And Isabel and Claudia managed to solve both of these structures using ARCIMBOLDO. But the point is that the first structure they solved was this second crystal form, so the slightly lower resolution, but just with a single chain. So they solved this using ARCIMBOLDO Lite, and we've heard about all of these things before, but so we all know that it uses ideal helical fragments in phasor. And then this is the important step, I think, the density modification and C-alpha tracing in shell XE, which can really improve things and really tells you whether it's worked or not. And an important point to make is that the correlation coefficient you get out of shell XE tends to be the best way of working out whether or not it's worked. Okay. And they use this, employing this coiled-coil mode, which includes fragment sliding to improve the sort of extension of helical tracing in shell XE. So this is what the structure looks like. So it's an anti-parallel tetrameric assembly, so in which you see from one pair of chains travel N to C in this direction, the other pair travel in this direction here. And that was solved from a shell XE correlation coefficient of just over 40%. So we had the other crystal form, and obviously we wanted to solve that too. So trying just molecular replacement, as you would expect, we've solved one structure, just molecularly replace it into the other space group. It doesn't really work. You get pretty good scores in phasor, but it doesn't refine. It's wrong. So Isabel and Claudia managed to solve this using ARCIMBOLDO Shredder. Okay, and we've heard about this a lot as well. But essentially, they took the template, which was the structure that had already been solved in the other space group, with the spherical model generation, put that through phasor, and then combined the phasors from a number of partial structures and density modification and tracing in shell XE. And this ended up solving nicely, correlation coefficient of 48%, and now you can see you get a pretty decent map and a pretty nice structure in the end. And the structure looks very similar to the other version, actually. There are a couple of interesting sort of differences, which I won't go into now, but it is essentially the same overall structure. And if you overlay them, they overlay pretty well. So 1.39 angstrom RMSD. So this really just highlights the point that coiled-coils are tricky. Even when you are starting with a model that is essentially the right structure, it still doesn't always work. And just from a biological perspective, just to explain why this was interesting. So once we'd solved this, we always work out that the crystallized form was actually the sort of self-assembled version, which assembles through protonation of histidine residues, and that this is a mechanism whereby SYCP1 self-assembles and then interacts with DNA within the meiotic chromosome axis. Okay, so then the other end of SYCP1 and the other method, so we crystallized the N-terminus of SYCP1, which we knew would sort of self-assemble somehow in the midline. So that's crystallized, gave a resolution of just under 2.1 angstroms, and that had 2 chains of 100 amino acids in the asymmetric unit. So we solved this through AMPLE, and this has, again, already been explained, but the thing I just want to really highlight here is that AMPLE uses a lot of ab initio models, and it's based on the principle that features that are common in a lot of models are most likely to be correct. So clusters and truncates, then uses a lot of search models through phasor, and then again it's this shell XE run here where you get density modification, and through auto tracing you get a correlation coefficient which essentially tells you whether or not it's worked. Okay, and as Dan explained yesterday, this can be run with quark decoys or with Rosetta models. And if you run it with quark decoys, you can run it on the online version. And if you run it using Rosetta models, you have to run it yourself. But the key point here being this Quark decoys are nice and quick and easy to do. Rosetta models, you have more input, so you can actually change things in some way, which can be important, as I'm going to get onto. And Dan put these up yesterday as well, so you can download the Rosetta fragments from this server, and then for Quark decoys, you can download them from this server, and you can run them on the CCP4 online server, which I would definitely encourage you to do because it's free from the point of view of your CPU. You don't have to do anything at all, so it's always good to run it that way. And this is how we solved the SYCP1-N terminal structure. So AMPLE will output a lot of jobs, and it gives you a lot of numbers from the RefMac, Buccaneer, the Phaser outputs and that. I would not look at anything at all other than the shell-XE stats, because that tends to be what tells you whether it's solved or not. And in this case, you have the shell-XE correlation coefficient. We had one solution with 35%, and everything else is 23, 22, 21. So one thing looked like it may have worked. What is that solution? Well, here is the shell-XE build, and it was quite fragmented, little bits of helix. But if you sort of look at it, you can see how it's sort of a coiled coil, isn't it? Okay, so we thought it may have worked, and the map wasn't too good. Yeah, there are indications there that it may have worked as well. Now, the next thing we had to do to be able to really build this properly was anisotropy correction of the data. So I said that coiled-coil proteins often have quite anisotropic data, and there are a couple of ways of dealing with this. There's the diffraction anisotropy server, and also there's Star Aniso, which runs in Autoproc. So we use the anisotropy server in this case, and it firstly diagnoses whether you have anisotropy, and we have severe anisotropy. It does get higher. I'm not quite sure what the statement is if it's 100, but anyway. So we have severe anisotropy, and then what it does is it does ellipsoidal truncation of the data to try to remove the data that essentially isn't there, the sort of noise. So in this case, it put it down to 2.1 angstroms in two directions and 2.6 angstroms in the third axis. But it's not just the truncation, it also does anisotropic scaling as well. So this scaling actually, I think, helps a lot in the downstream processes. So with anisotropic correction, we got better maps, but importantly, we got better builds, which was really the key. And with a lot of building, we managed to build the full structure and refine it. And when I say a lot, I think it took a week before we really knew what the structure looked like. So it did take a lot of iteration here. And this structure is up here. So it's a dimeric coiled-coil that interacts head-to-head in the midline. So it essentially forms a dimer of dimers structure. And you can sort of see that within the context of the wider SYCP1 molecule. So the structure we solved is here. So you can see the head-to-head. It's how the two chromosomes are pulled together in the midline. It's sort of how the zipper works. Okay, so that's the sort of biological story. But now we do a bit of development of the methods. So I've shown that AMPLE worked in that case, but the solution was pretty borderline. And what we noticed was that if you look at the models that we're trying to solve this with, the models are pretty terrible, because we're dealing with a coiled coil, and we know that a coiled coil is elongated and oligomeric, whereas all the models that we start with are monomeric and trying to form sort of globular folds. So the sort of thought was that by the models being created in this way, we're sort of biasing it against ourselves, aren't we? So we wondered whether we could create more realistic models, and then maybe we could enhance the ability of AMPLE to solve coiled-coil structures. So we firstly did this in a very, very inelegant way, but it actually worked, which is sometimes what happens in life. So what we realized was that in Rosetta, when Rosetta builds models, it puts a penalty on non-globularity. Okay, so you can simply switch off the RG penalty when you run Rosetta through AMPLE. So that's the first thing. The second thing is that we know what a coiled coil looks like. A coiled coil has a long helix. We know the size of a helix. A helix is around about 1.5 times the number of amino acids in angstroms. So also, when running Rosetta through AMPLE, we can impose a distance constraint where it basically says that the chain has to be a certain length from its end to its C terminal end. And we can just work that out on the basis of the length of the chain. And when doing this, you still generate pretty rubbish-looking models, but they're pretty rubbish-looking models that have long helical structures, which are a bit more like what we want. So they still look pretty sort of rubbish, but it's a bit closer to what we're after. So we tried this with the same SYCP1-N terminal data set, and we found that it works a lot better. So this is one of the models that was generated for it. But here are the SHLX-E correlation coefficients, and these are all above 40. So we now have a lot of runs this worked in this case. And and then if you look at what worked, so the top solution, Phaser placed 2 helical search models, and then SHLX-E was able to build something that looks a lot more like a coiled coil than what we saw before. It's still not complete by any stretch, but it looks better, and you can see from the map that it's obvious as to how you can quickly build in extra sequence. And here it is in the context of the dimer of dimers, just so you can see sort of the bits of coiled coil that are missing. And so we wondered whether this could be generally applicable. And we took a couple of test cases and just ran it side by side. So Rosetta using the defaults and Rosetta using the new flags for the elongated structures. And in one case, we were able to get the successful solutions up from 30% to 64%. And in another case, it went up from 21% to 79% of the runs working. So this is sort of promising as a new way of approaching coiled coil structures. And what would now be nice to do would be to test this over a wider set, including ones that don't work at all normally, to see whether these elongated models can get around this problem. Okay, so that was a pretty inelegant way of dealing with the problem. We wanted to do something fancier. So what we thought was, can we just build a coiled coil and use real coiled coils as models that get put into AMPLE? And to be able to do that, we need to know what the coiled coil looks like in advance. Is it a dimer, trimer? Is it a parallel coiled coil or antiparallel coiled coil, etc.? So something I just want to point out is that in a lot of our other work, we have really been building up methods where we can work out the structure of a coiled coil from the solution experiments. So we don't necessarily have to rely on the crystal to actually find this out. And just briefly, I'll just explain that very straightforwardly, helical content, you work that out with circular dichroism very, very easily. Oligomeric state, size exclusion chromatography, multi-angle light scattering will give a completely unambiguous determination of whether you have a dimeric coiled coil or trimer or tetramer or whatever. And then small angle X-ray scattering is extremely appropriate for coiled coils because they're long and thin. They give unusual scattering profiles, and you can use the real space P of R distribution, which you work out from the Sachs scattering, to determine the length of a coiled coil, because the length of a coiled coil is simply the maximum dimension within the structure. So it's a really, really easy thing to work out. And you know the theoretical length of a helix, so if that matches the length you get here, you must have an extended coiled coil. If it's half the length, then the helix falls back on itself. So it's pretty easy to work these things out. From the Sachs data, you can work out the radius of a coiled coil, and the radius tells you how many helices there are. So in this case, if it's 8 angstroms, it's 2. If it's 10 angstroms, it's 4. So you can really build up the structural information. And the final thing is which way around it goes. Is it parallel or anti-parallel? So we found that using Sachs, putting an MBP tag on the front of the protein, we can determine whether it's parallel or antiparallel, because the interatomic vectors between the MBPs dominate the SAC's scattering and show up as peaks in the real space distributions. So we're able to work out in advance now if something is a dimeric, coiled, tetramer, or whatever, parallel or antiparallel, and its overall shape. So can we put all of this into Rosetta to build models of what we expect to get in our crystal, and then can we use that to solve it? And the answer is maybe, or, well, yes-ish, I think is the answer, actually. So we can do this using Rosetta fold and dock. This is a module that it builds just a single chain, but with symmetry operators. Okay. And what we input is we input symmetry operators to tell it the number of chains and the helical orientation, and distance constraints. This is the really important bit. That will determine the orientation and the overall shape. And we had to play around a lot to work out how to get this to work nicely. And here are two examples. So a parallel dimeric coiled-coil. Here's an example of a Rosetta model that we made with that. And if you can see how it's picked out this really nice sort of stretch of what's essentially a leucine zipper region. And something that I just want to point out here is that I'm not using easy coiled-coil sequences. So this is a coiled-coil that exists as a dimer or a tetramer, depending on the pH. So I'm using a case in which, looking at the sequence, you wouldn't really have a clue what type of coiled-coil it was. And, well, you can't really make out a heptad. So Rosetta's working it out for us, which is the whole point. And then we can also get this to work with antiparallel structures as well. So we can form nice antiparallel tetrameric structures. And you can see, just looking at the local folding, it's picking out some sort of relatively nice hydrophobic cores. And just one thing to point out, the method we've got working is pretty computationally inexpensive. 15 minutes for each model, so on a decent desktop computer, you're talking about less than a day to generate 1,000 models, which you would need for an AMPLE run. And over 25% of them are good as well. So it's not as though we're generating a lot of rubbish. So we thought, well, can we use this to solve a structure? And the answer is yes. So we did this again with the SYCP1-N terminal structure. So we built a number of these dimeric coiled-coil models, and then we got some nice clear-cut ShellXE scores which show that we have solved it. If we look at the top solution came from this was the search model this phaser plays. So you can see it. We've done what we wanted. We've used a coiled-coil as the search model. And what's nice in this case is that ShellXE built essentially the whole structure without having to do anything else. So something about this has worked a little bit better. And as you can see from the map, it has these rather enticing green blobs waiting to have side chains put in. And there's the full structure, just so you can see a little bit of extra building in the midline. So, I mean, this is just a single test case, but it at least provides proof of principle that it is possible to generate oligomeric coiled-coil structures and use them in AMPLE to solve a structure. And it would be nice to see if we can extend this. So just to finish, I just want to sort of summarize the methods that we've been talking about. Essentially, AMPLE and ARCIMBOLDO should be able to solve any coiled-coil problem that you have. And this new thing that we're sort of thinking about is making these these elongated Rosetta models to put into AMPLE, and also these oligomeric coiled-coil models, which we've initially used for AMPLE. And I sort of thought during the talks that maybe they could be put into ARCIMBOLDO shredder as well. So that's something that we can talk about. And the final, just a little few comments to make is that if none of this works, go back to the data, and the things that we find are data sets. One data set works, another data set doesn't work for whatever reason. Resolution, obviously, the better resolution will normally help. The space group is often wrong with the coiled-coil, or at least you can get the screw axis wrong pretty easily. So I would try all of the possible space groups. Any similar data sets, merge them together can sometimes help. Anisotropic correction, this actually can help to solve the structure. So doing anisotropic correction before doing all of this can actually be the difference between it working and not working. Okay, so finally then, so thank you to James grew the crystals that I've used in the talk. Guru did some of the scripting. Isabel and Claudia ran ARCIMBOLDO and solved the C-terminal structures. And Dan, Jens, and Ronan helped with AMPLE. So thank you very much.